Good evening, everyone. I'm Graham MacDonald from the National Stem Cell Foundation, and we're very proud and privileged to be sponsoring this public forum on stem cells and current research, and we hope there'll be lots of questions and participation on your behalf. It is a great privilege for me to open this forum. The foundation was set up 7 years ago, and its aims were, as you've been watching the slides, to promote stem cell research, to educate the public about stem cells and their use, and to provide funds for research and to capitalise on the finding of stem cell science to bring solutions to unsolved health problems, principally in man, but as someone points out, probably there'll be veterinary applications as well. When we started the foundation, we answered a lot of patient queries about stem cells, and these were mainly about where people could receive stem cell treatments, problems they were having with stem cells, and we were in a very unfortunate position that we almost always had to warn people that most stem cell treatments at that stage were unproven. But as you know, if you take a medicine, it's gone through an extensive regulatory and clinical trial period, and most stem cell treatments, in the case of most stem cell treatments, that hadn't occurred. So one of the important initiatives that the Foundation is embarking on as I speak is to sponsor 3 major clinical trials that will give us definitive evidence for the use of stem cells and cellular products in 3 major and very different disease areas. I think at the moment I can't actually say what they are, but this information will be released when we finalise our contracts with the research groups. We are providing funding on a dollar-for-dollar basis, with the Foundation contributing $1 million over 5 years. And this is realistic funding over a realistic term to give a proper outcome of clinical trials. So I hope we'll be able to make better use of this in the future. But to come back to the idea of stem cell treatments, one of the things, and I'm sure this is going to come out in what you're about to hear now, is the very significant advances in stem cells and their uses. And this is based on our understanding of the biology of stem cells, but also supported by a lot of other science. And if I can just mention one aspect in particular, one science in particular, there is a technology called CRISPR, C-R-I-S-P-R. I won't bore you with telling you what the letters stand for, mainly because I have difficulty remembering them. But CRISPR is a way of editing genes. So if people's cells have a faulty gene, and we know all about various diseases which are sheeted home to the faulty messages that cells are receiving from their genes, CRISPR is a way of editing, of precisely correcting the defect in genes. And the reason I bring it up is because it's easy to think of stem cell science and stem cell scientists as leading us towards cures and definitive treatment for diseases. But what we must never forget is that all of these things are founded on a very deep column of science. So, for instance, CRISPR was discovered in about 1987 by a Japanese research worker who had absolutely no interest in stem cells whatsoever. It was a way that bacteria protect themselves from virus infection. It was about 10 years before somebody realised that it could be used to program cells. 2 research workers in Spain worked out what the intermediate steps were, and about 2 years after that, coming into the 90s, coming into the 21st century, some American workers actually worked out a technology for doing it. And the reason I mention it, that all of that work was done on biology and biological ideas that had been developed over 2 centuries. And the tools they used were based on science, and they were based on physics and mathematics and chemistry. And so the really important thing to remember is that for us to realise the benefits that we know we can get from stem cells will be a work of high science. And at the moment, we're investigating hope and imagination and incredible commitment, as you'll hear from the speakers later on. And it will be science that gets us to the point where these findings that are very exciting to scientists, sometimes hard to explain, will become available and will become useful to the public and all of you who are here interested tonight. We've got a panel of experts, which you've been seeing on the screen, and I'd like now to introduce Associate Professor Megan Munsey of the University of Melbourne and Stem Cells Australia. She was very cagey when I asked her what she actually did, but I can tell you that she is a card-carrying stem cell scientist with many years, like over a decade's experience in advocacy, in the ethics of stem cell research, and I think she is absolutely the ideal person to introduce the speakers and to run this show. Thank you, Megan.
For that lovely introduction, and thank you everybody for coming along tonight. It's a fantastic crowd, and I want to thank the Foundation for helping us have such a beautiful venue. I think what we're going to talk about is some lovely cutting-edge science in a gorgeous cutting-edge sort of architectural building, I think. So thank you very much everybody who's come along tonight. And I think you're in for a bit of a treat. Obviously, as Graham mentioned, I've had a long interest and a passion about stem cell research. And what we want to do tonight is share with you how we see stem cells, how we use stem cells in our research, and what we see the future that stem cells could contribute to. Most importantly, we wanted to have tonight's event as a kick-off of a celebration of a week of stem cell science. We're absolutely delighted that Melbourne is the host of the International Society for Stem Cell Research's annual meeting this year. It's a real honour to have the meeting here and to be joined by over 2,500 researchers, scientists, doctors, policymakers, bioethicists from over 50 countries who've come to Melbourne to talk about their research, to share their latest breakthrough, to talk about some of the challenges in taking discoveries from the bench into the clinic. So it's a really exciting week for us, and I want to give a shout out and ask you all to put your hands together to welcome some of our international visitors who I know are in the audience. I wanted to give them a big warm welcome to Melbourne because, of course, they've come mostly from the Northern Hemisphere where it's a bit warmer than Melbourne at the moment, but we're delighted that you're here. And of course, tonight's about you. So while we're really thrilled to have so many stem cell scientists in Melbourne, we really want to talk to you about where stem cell research is at. There was quite a disturbing, at least from my perspective, survey done a couple of years ago that asked Australians what their attitudes were towards various aspects of biotechnology. And when people were asked to kind of reflect and to acknowledge whether they were aware of stem cell science and other aspects of biotechnology, stem cell science was kind of a recognised term by over 90% of the people who were surveyed. But when a follow-up question was asked whether they could perhaps articulate or explain what stem cells were to their friends, only about a third could. And we want to change that tonight. We hope that after tonight's presentations and the opportunity for you to ask questions, you'll know a little bit more about what stem cell research is, what stem cells do in the body, and possibly the applications of stem cell science for the future. So before I introduce our esteemed panel, I thought I should just also introduce some key terms. Now, you're going to hear this again and again, perhaps from each speaker, and don't be shy if you need to ask again. But I think it's really important that we acknowledge that the word stem cells is thrown around quite a lot, but perhaps we aren't clear enough about what stem cells do and what they are. So I thought it'd just be worth perhaps at least giving my perspective, and my colleagues might have a slightly different perspective, but from my perspective, I like to think of the fact that our body is made up of different organs, and each organ is made up of different tissues, and within those tissues are cells. Stem cells are a special type of cell that can give rise to cells of that tissue, of that organ, but they can also renew themselves, so they can make a copy of themselves. So when I think about stem cells in this context, I'm thinking about tissue stem cells. And these are stem cells that really keep us healthy. And perhaps the best example of a tissue stem cell is a blood stem cell. And you're probably familiar with the fact that people, or that your blood stem cells make your red blood cells and your white blood cells and your platelets. And perhaps you're even familiar with somebody who has leukemia who might have had to have a stem cell transplant. These are all examples of how we can already use stem cells in the clinic. What if we could extend that? What if we could harness the potential of stem cells in other organs to repair damage, to heal someone who's got after an injury, for example? There's another type of stem cell that you might have also heard about. Perhaps you've heard about stem cells made from human embryos. We call them embryonic stem cells, and these are slightly different in that they're isolated and made in the laboratory from donated IVF embryos, and they have a capacity not just to grow into one particular cell in the tissue, but into many different types of cells, provided they're given the right signal. This discovery was made about 20 years ago, and you might also be aware that there's another type of very similar cell called an induced pluripotent stem cell. And this discovery was made about 10 years ago and has really changed the field. Both these types of stem cells can be grown in large numbers in the lab, and then given the right signals can be grown into a specific cell type. So this has really changed, I suppose, the repertoire of what scientists can do in a lab. And these new type of cells, these induced pluripotent stem cells that can be made from a patient's skin cell, can actually now let us discover and understand a disease in the laboratory, effectively grow a disease in a dish. And you're going to hear some examples about that tonight. So that's what I wanted to say about background. I also wanted just to make a comment that obviously over the last couple of decades we've made incredible progress in the field, but stem cell research is also, like many other areas of science, is also surrounded by controversy and presents challenging ethical issues. And I hope we'll get to discuss some of those tonight as well. So I'm going to now call on my panel members to come up, and I'd like to start with Professor Susie Nielsen, the CSIRO, and from Australian Regenerative Medicine Institute at Monash University. Susie is a stem cell scientist who works with blood stem cells to understand their role in diseases affecting the blood. Our second panellist, all the way from Western Australia, is Associate Professor Pratinder Kaur from Curtin University. Pratinder has long been interested in stem cells found in skin and their role in wound repair, ageing and cancer. Coming to us from Sydney is Dr. Michael O'Connor from Western Sydney University. Michael uses stem cells to learn more about the causes and potential treatments for cataracts and other eye diseases. Michael's a past president of the Australasian Society for Stem Cell Research. Also from Sydney is Associate Professor James Chong. James is from Westmead Institute and the University of Sydney. James is a practicing cardiologist and leads a cardiac regeneration laboratory at the University of Sydney. Our final panellist is Professor Melissa Little from the Murdoch Children's Research Institute and the University of Melbourne. Melissa heads a team of researchers who use stem cells to recreate kidney tissue in the laboratory in an effort to better understand kidney disease and improve diagnosis and treatment. Melissa is the president of the Australasian Society for Stem Cell Research. She's also the program leader for Stem Cells Australia, which is the Australian Research Council Special Research Initiative in stem cells. And Melissa has played a leading role in organising the Melbourne International Society for Stem Cell Research meeting this year. So please join with me in thanking our panellists and welcoming them. Thanking them for coming. And what we're going to do now is we're going to hear from each of the panellists in turn. They're going to tell you a bit about how they use stem cells in the research and why it matters, why their research matters. At the end of hearing from each of them, we're then going to turn the floor over to you. So while they're speaking, if you think of a burning question, jot it down, make a note, But please raise your hand at the end and share your thoughts and any kind of queries you have. So without further ado, Susie.
So thank you very much, Megan. It's a great honour to be here to talk to you today, and I'm very lucky because I get to discuss my favourite topic, which is your blood and bone marrow stem cells. So before I start talking about the kind of stem cells I work with, I wanted to further cement what Megan was saying before about the different types of stem cells, because if everybody could take home one understanding today, it's a better understanding of stem cells and what the word stem cells actually means. So as Megan alluded to, we have the new kids on the block, which are our pluripotent stem cells and our embryonic stem cells. So Megan already told you that these are relatively new area of scientific research for stem cells, less than 20 years old. But they have the hope of being able to regenerate any different type of organ in your body, and hopefully in the future we'll be able to do this in regenerative medicine and use this. However, at the moment, these stem cells are largely restricted to being able to make specific organ cellular types in a dish, and most of the research using these types of stem cells is still in the laboratory. And we are yet to realise the hope and the potential that these types of stem cells can make a whole new organ that can be then used in the clinic. The stem cell that most people commonly forget exists and has been used in the clinic for many years is the other type of stem cell, the adult organ stem cell, as Megan spoke about before. And the classic example of the most commonly used stem cell in the clinic is your bone marrow stem cell, otherwise known as a hematopoietic stem cell, where hematopoiesis actually means blood. So these are the stem cells that live inside your bone marrow, and they can generate all your blood cells and do so every second of your life. And these are the types of stem cells that I've worked on. And I'm very fortunate because Australia has a very rich history in contributing to our understanding of blood stem cells and their use within the clinic. So what I'd like to get across is your blood stem cells are very important because you're required to make 1 million mature cells per second every minute and every day of your life in order for you to survive. And as a consequence, really requires tight regulation of the stem cell that gives rise to all these cells in a tree-like fashion, as you can see up on the screen. And my research has really been trying to understand the environment in which these stem cells live in and how that environment educates these stem cells to mature and divide when required in a normal situation, and then what goes wrong in the situations such as leukemia, and how can we then best modify treatments in order to be able to treat this. So speaking of Australia's rich history in terms of blood cell stem cells, stem cell research in this area started many, many years ago. In fact, in the late 1950s, it was first discovered that stem cells or bone marrow could be used for treatment of cancer. It was then two fantastic eminent scientists, both from Australia, Ray Bradley and Don Metcalf, that significantly contributed to this field with their discovery of certain growth factors that eminently were involved in terms of the regulation of hematopoietic or blood stem cells. It was their pioneering work that allowed us to isolate a growth factor, again from another Australian researcher, Nick Nicola at the WEHI here, together with Don Metcalf, who allowed scientists to then use a growth factor, GCSF, which is absolutely essential in terms of not only stem cell regulation, but in terms of the clinical isolation of stem cells today. It was a couple of years later that the human form of this growth factor was isolated, and then clinical trials using this for mobilising stem cells occurred and was founded from a group from Adelaide a few years after that. So in this context, mobilisation is a new process which allowed clinicians to entice the stem cells to leave your bone marrow, go into your peripheral blood, and therefore they could easily isolate these cells without having to use general anaesthesia and an operation to isolate these cells directly from the bone marrow. This was a huge advance in terms of clinical use of stem cells, and this was many years ago now. So the first successful transplant using stem cells mobilised in this fashion was in the 90s. And then GCSF became licensed for clinical years a year or two later. And more recently, there's been advances in this process because, as everything else, there's new discoveries made. And now, rather than having to use a growth factor alone, they can use small molecules that can add to this. And what I'm about to tell you in terms of our very key interest in this area is adding to this as well, where you can take a very small molecule, you can use that together with the growth factor or on its own to entice these stem cells to leave the bone marrow and enter the peripheral blood where they can be easily isolated for clinical use. And this small molecule was now approved about 10 years ago now for use for transplant in terms of mobilising stem cells. Now, whilst all this clinical development's been going on, there's been a lot of before I move on, sorry, I meant to tell you that the latest things or the newest things that we believe will be coming in this space in the next few years, and they've been all over the news, so I'm sure most of you have already heard about them, are treatments such as CAR T cells, immunotherapy, and gene therapy in this space as well. Now, whilst all this advances in the clinic's been going on, of course, it's underpinned by a lot of basic research in the laboratory, our understanding of your bone marrow. Again, this has really been matching our movement into the clinic, where the first understanding of how important the microenvironment in which these stem cells live in happened back in the 70s, where the first description of a niche, which is the environment these stem cells live in, was first hypothesised. We then understood that you could take one single bone marrow stem cell and you could recapitulate the entire blood system from that one stem cell in an experimental situation. This is really important because it tells us the power of these stem cells. More recently, we've identified where that niche is located, and that's critical for the research that we're doing because understanding where a stem cell lives allows you to understand what regulates it, and by understanding what regulates it, allows you to improve that kind of clinical output. Where is this research heading? We need to understand more about the specific types of cells that regulate the stem cells in your bone marrow, and many, not only blood cells, but extramedullary molecules, the microenvironment, nerves, blood vessels, all of these things play key roles, and we need to understand these roles if we wish to take this further. So in terms of my research and where I'm specifically interested, we've recently identified a small molecule, which we have termed BOP, which we're currently taking into a phase 1 clinical trial. And BOP has the hope in 3 specific areas in terms of stem cell research. Not only can it work with this other small molecule that I just mentioned to you before to mobilise stem cells, but it can do it in one injection in 90 minutes, which is a huge clinical advance where you don't have to have multiple repeats of a growth factor and an ongoing procedure. So that allows a far more efficient process. It allows people who didn't mobilise before to mobilise now. This small molecule also has a promise in chemosensitization. Chemosensitization is, unfortunately, in disease states. Some of the disease cells remain dormant in the marrow and can contribute to relapse at a later date. Our small molecule can dislodge these from the environment, allowing them to become sensitive to chemotherapy and hopefully then remove them, allowing the disease to be better treated. And finally, our small molecule has the promise to be used with gene therapy. It allows isolation of cells that can be genetically corrected, that then can be retransplanted back when the disease involves the blood system. So I'd just like to leave it there and hand over to our next speaker.
Okay, so it's time to turn from the blood to the skin, my favorite organ. So for the first slide. So some of you might think that space is the final frontier, but I'm here to tell you it's actually your skin. So why do I say that? That's because basically without your skin, as this unfortunate man over here appears to be, you are vulnerable to dehydration, and you're vulnerable to pathogens invading you, including bacteria and fungi. And so these are life-threatening functions that the skin performs that keep you alive, right? And so in terms of clinically, this is the first problem we addressed, that we try and focus on trying to give back these cells that form the barrier. So actually, if you look at skin, you'll see it's quite a deep organ, and there are 2 kind of compartments, if you like. So the top one, which is shown in high power just to the middle there, is the epidermis. And the job of this tissue is to generate these differentiated cells or squames that you see in the top right. And this is what keeps you watertight and impermeable, basically. So my lab has been spending some time actually trying to understand where the stem cells of that epidermis are, and they're in that sort of bottommost layer of the epidermis. And so our sort of claim to fame, in the past anyway, is that we were the first to isolate skin stem cells from human skin, and then we could put them through their paces to demonstrate that they did indeed have the ability to regenerate huge amounts of skin tissue. So the next challenge though is that although we can replace this epithelium and keep you alive, there are a whole range of functions that are still missing when you get transplanted after you've lost a lot of skin. And so these really involve the tissue that's sitting underneath the epidermis, which is the dermis, this nice thick stuff, keeps your skin nice and juicy and plump. And there actually is a variety of cell types that exist, and we don't really understand a lot about this particular tissue, much less the stem cells that are in there. So the organs that are present within this whole skin then do things like control your temperature. This is kind of important if you're in the Australian outback or even otherwise. Oil secretion, so sebaceous glands, this is missing from the transplants that you get. And so you have very dry skin. Sensory function, so the thing that tells you, take your hand off, it's too hot. So we're missing the innervation that is present in your skin from the transplants that the patients get. Subcutaneous fat to keep you warm in the cold Melbourne winter. And then the blood supply as well is actually not given back, right, to the patients. And that's simply because we actually don't know how to do this just yet really well in the clinic. But what my lab is also starting to realize, and this is hopefully our claim in the future to fame, is that these cells that are sitting around the blood vessels, we call them pericytes, they actually have stem cell-like functions as well, and they can regenerate a lot of tissue. But like Susie was mentioning just now about the microenvironment, they seem to be a really critical component, and they speak to the epithelial cells sitting on top, secrete goodies that say, hey, stay like a stem cell and don't leave, right? Because most of the other guys are just sort of going through those layers, making dead cells, and then lost to the environment. So we're hoping that we'll have some more progress there for you. Now, Megan wanted me to tell you a little bit about this really heartwarming story involving the use of stem cells of the epidermis in the clinic. So this is really kind of a story that will grab all of us. My apologies for showing you nasty things before you have your dinner. But in the top left, you can see a picture of a boy who was 7 years old, and he was admitted to a hospital in Germany. He was born with a mutation in his skin, basically in the epithelial cells, which prevents them from attaching to the dermis. So it doesn't take a lot for him to blister, right, over his entire body. So he developed a bacterial infection, and by the time he was admitted to hospital, as you can see in the top left, the red bits in that cartoon are to show you which parts of his skin were missing, actually, his epithelium. So he had about 60 to 80% of his epidermis was gone, right? So he basically was facing certain death. So what these guys over here did, so this couple that you see are Michela De Luca and Graziella Pellegrini. They are colleagues of mine, but also friends, as we should all be when we work in the same arena internationally. They've been working for a long time to grow really well these epidermal cells in a dish. This is not really a routine thing, it's an art form. So what they did was they took the remaining healthy cells from this boy and they put them in culture, and then they genetically modified them to introduce a correction for the gene that was mutated in that skin, which will now allow those cells to attach, okay? So then what they did was spend a lot of time, months, growing up these genetically modified cells, verifying them, making sure everything is cool, and then grafting in a kind of patchwork all over his body over 3 separate operations. And the good news is that after 8 months of this treatment, this boy is back at school and he's playing soccer, right? It's a great outcome. However, there are, of course, some significant challenges that are left for this boy and for other patients. And that, as I alluded to earlier, is basically that you're not really replacing all the other functions that skin needs to have for you to have a normal life. But these kinds of patients also have a problem where the lining of their esophagus, so the feeding tube, basically also kind of peels off. And you can imagine it's not so easy to do patchwork, on the tissue that's inside. So we've got to come up with clever ways to help these patients. So the other thing is that we're using, at this point, mouse feeder cells to grow the epithelial cells. And really, in terms of clinically, this is not a good way to go. So another spot where my lab is trying to get around this is to use human pericytes, which we think actually are very good at supporting epidermal growth, and then be able to use that rather than a mouse cell type to do that. There's another issue when we introduce a gene into a patient, and that is that you actually, using the technique these guys used, and remember this is a compassionate use of this technology that's really experimental, is that you randomly introduce the gene, the corrected gene, into the DNA in several places. And therefore you run the risk of perhaps inserting it in a place which is not good for you. In other words, you turn on a gene which is now going to cause cancer. So what would be really cool, as Graham said earlier, was to be able to target that using CRISPR-Cas technology and replace the mutated gene with the normal gene. So you just get, switching happening. So that's something we can work on, and certainly restoring dermal tissues, things like hair follicles and sweat glands and so on, so we can have people who don't just have barrier function, but actually now have skin that stretches and skin that can cool itself down and all the other things that I've alluded to. So on the right, you're seeing some pictures generated from my lab at the bottom right, which are asking actually the very same question that Susie's asking in the blood, in the bone marrow, which is that we know there's an immense crosstalk between the dermis and the epidermis, and this is not idle chatter. This is actually important for the stem cells to renew themselves at the top, and perhaps even to renew the entire dermis, and that's where I hope that my lab will focus its efforts in the coming years. So thank you.
So I appreciate the opportunity to tell you a little bit about my work tonight, but really a journey from a whole lot of researchers around the world that spans the entire scientific journey from basic discovery through to clinical translation that ends up in a new treatment. I'm going to tell the story in reverse though. And the system that I'll be talking to you about is the eye. And we can see in this diagram up here that the eye allows us to interact with our environment. Light comes from the environment. It enters that first tissue there right on the outside of the head called the cornea, which most of you, I imagine, are aware of. It provides about 70% of the focusing power of the eye, and it focuses the light onto the lens, which is that yellowy tissue right just behind the cornea. And the lens provides about 30% of the focusing power of the eye, which allows the light to be focused onto the retina, which is where you see the image of the flower at the back of the eye. So I'll talk to you about each of those three tissues, and we're going to walk our way back from a clinical translation through to basic research. So starting off with the cornea. So the cornea is built up of cells, just like Megan mentioned earlier. Like bricks in this building's made up of bricks. It provides a structural framework. Our stem cells, as Megan mentioned, imagine there's a brick that can make other bricks and make itself as a new brick. This is what happens in our tissues, including the cornea. And so if you see these little green dots here, they're called limbal stem cells. They're a very special type of cell that's on the very outside of the cornea that can make that front surface of the cornea. And when we're all healthy, light comes through the cornea and we can see this image like the two boys there holding a ball each. But if we get a burn of the cornea, imagine that an acid or an alkali spills into our eyes, the surface of the cornea becomes no longer transparent. We lose the ability to see. So in that situation, a pair of researchers about 20 years ago now discovered that if you take some of these limbal stem cells, these green cells, and you take a biopsy off the patient so that their own cells are being used, and you culture them in the laboratory to expand their numbers, you're able to then transplant them back onto the surface of the eye, and that restores the vision in the patients. So that initial study was done or published in 1997. It was only 2 patients at the time. And between 1997 and now, that additional research has gone on into that particular process to optimize it, make sure that it does work on larger scales, and it's now one of the really 2 main accepted types of stem cell therapies. And the people who are responsible for that, the original publication, were Graziella Peregrini and Michelle De Luca that Pratinda had mentioned just before. And their work is about to be recognised at the international conference here in Melbourne through the ISSCR's Award for Innovation. So both science-changing and life-changing research there. So the next tissue of the eye, that middle tissue, is called the lens, and that's the part that my group works on. And particularly as people age, but babies can be born with this condition as well. Cataract is when the light isn't able to go through the lens, but it gets blocked. And so the image of the flower is no longer visible at the back of the eye. And when that happens, it affects vision to cause low vision, and it can progress all the way through to blindness. It's very difficult to get access to lens cells. Limbal stem cells, you take a biopsy off the front of someone's eye, but for the lens it's not possible really because it's inside the eye, and no one's going to donate that tissue for research. So my group was interested in working out how we might be able to make large numbers of lens cells in the laboratory to work out how we might be able to better treat cataract. So we use a form of stem cells called pluripotent stem cells that Megan mentioned right at the beginning. They can make any cell type in the body, and we can culture them to make very large numbers. So it becomes a source of normal or diseased human cells. So my group built on the work of others, a lot of other people, and what we were able to do was find a way to take our stem cells and turn them into a lens cell, a human lens cell, and do it in very high purity. We then thought, well, in order to understand cataract, particularly age-related cataract, we need to have a lens that focuses light, something that we could work with in the laboratory, but that performs the same functions that the lens in the eye does. So we found a way to make little balls of these cells and grow them in the laboratory so that they gradually became transparent, and they accumulated the ability to focus light. So that set of 4 images that you see there, the top left-hand side is one of these microlenses that we make in the laboratory. It's relatively transparent, but underneath it, the picture, the really bright spot of light that you can see is it acts as a lens. It can focus. We tested it in many ways to show that it's got the lens cells. The lens cells have the right proteins, the lens cells are the right organization, that they're undergoing the right processes. So it gave us some confidence that we might be able to model a disease like cataract in our system. So the other images that you see there, the top right-hand side, you see one of our little microlenses that is no longer transparent, and if you look at the image underneath it, it no longer focuses light. And that's because we exposed it to a drug that's suspected of causing cataract in patients, and in our system here, we're able to reproduce that. So for the first time in the world, for the first time anywhere in the world, we're now able to replicate all stages of cataract formation in the laboratory in a way that's not possible to get access to that same type of tissue in a human patient. So the idea is, well, if we start to screen this system and we find drugs that can delay or stop the cataract formation, then we might be able to give them to patients as an eye drop. Or in certain situations where that might not be possible, such as an infant who might be born with cataract and you can't delay the formation of the cataract because it's already there, what we might be able to do is transplant some of these lens cells into the eyes of the children and see if we can regrow a lens so that they have an improved treatment. So experimental, but encouraging results so far. And then the final part of the eye that I'd like to talk to you about is the retina, there at the back where you see the image of the flower. So the retina is a much more complex tissue than either the cornea or the lens, and in the bottom right-hand corner you see the little colourful diagram that gives you some idea of what the cell types are and what their organisation is. The ones that sense the light, the rods and the cones, they're actually in the middle. Those coloured cells in the middle there sense the light, they turn it into an electrochemical signal that goes to the nerves, and then the nerves take that signal all the way to the brain, which is that big blob that you see on the left-hand side. And then behind the retinas, behind the photoreceptors, are a cell type called the retinal pigment epithelium that help to keep the entire environment healthy. So again, when everything's working well, we can see the images of the boys. But one example of a disease that happens in the retina, and it's a disease that probably many of you are familiar with, macular degeneration, age-related macular degeneration. This might be an example of what a patient sees when they get macular degeneration. They lose that central part of their vision. So my group works on this, and we do a little bit of work on it, but there are some world leaders that are here in Melbourne, so Elise Peabay and Alex Hewitt, who work in a much broader way on the eye. But one of the things that we do is we make a retinal pigment epithelium. So those cells that are on the left-hand side of the photoreceptors, they're black cells in the eye, and their job is to absorb stray light and to do a few other things. So we can, again, use our pluripotent stem cells that can make any cell in the body. We can use them to make these retinal pigment epithelial cells that we see them in the dish. And Elise and Alex, they're interested in looking at the broader sense, the broader retinal cell types that you find in the eye, and they're developing methods not only just to make these other cell types, but they're also generating very large resources of patient-specific disease cells, of disease pluripotent stem cells. So if someone has a particular eye disease, they're able to take skin biopsies, turn them into pluripotent stem cells, try and make these cell types that are present in the eye in order to do disease screening and drug screening. So the idea, the hope that we're working towards is that using these stem cell-derived cells, we might be able to make cells for transplantation or find drugs to use as therapies, or potentially down the road on that right-hand side of the green line, the DNA inside a capsule is the idea of gene modification technology, that these cells might be used as a vehicle for introducing treatments through modified genes within the body. So thanks for listening, and I'm going to pass over to James.
Thanks, Michael, and thanks to Megan and everyone who's put on this fantastic event. I'm really pleased to be able to be part of it. So my name's James Chong. I'm a cardiologist and also a stem cell researcher. And the brief was to introduce a little bit about a bit about my research, but why it matters. And if we take the second part first, the bottom line is that heart disease or cardiovascular disease is the biggest killer around the world. There's a statistic up there, 1.4 million people in Australia are suffering from heart disease, but that's probably an underestimate. And then my research is looking at stem cells or regenerative therapies to try and fix, in particular, heart failure. And heart failure is a condition where, as you as it might be suggested, the heart fails to do its primary function, and that is to pump blood around the rest of the body, to give oxygen to all the organs in the body. And so that statistic up there is saying that 1 in 2 patients with severe heart failure will die within a year of diagnosis, which is a worse prognosis than many forms of cancer. And many people don't realise that. And I have to point out that that's severe stage heart failure. There's in the early stages of heart failure, the prognosis is a lot better. A number of things can cause heart failure, but heart attacks are probably the most common. And I bet many of you, if not all of you in the audience, will know a friend or a family member who has had a heart attack. And so 350,000 Australians, they say, suffer a heart attack at some stage in their life. And again, I think that's an underestimate. It's probably a little bit more. And the last statistic is that costs a lot of money. And not just money, a lot of problems in lost productivity and lost loved ones. So that's why I feel my research is important. And why does all of this happen? Well, the heart is one of the least regenerative organs in the body. When I was in medical school, I was taught that when the heart injures itself, it can't repair itself. So when I found out that there were stem cells or an ability to be able to reverse that, I was obviously very interested, and that's what I've been dedicating my research interest towards. And you're probably wondering why that little guy on the left of screen is there, an axolotl, or a Mexican walking fish. So they have an ability to be able to regenerate things like if you cut off their limb, they can regenerate it. And we've worked out that if you cut off part of their heart, they can even regenerate that too, which is really quite phenomenal. That sets up a precedent then in biology of this heart regeneration that hopefully we can use and take into therapeutics for my patients. So, and that brings up the middle picture. Broadly speaking, that could take the form of two strategies. We could either harness stem cells that we believe reside within the heart itself and make them do the job that they're currently not doing. And that's adult stem cells, as has been introduced. But unfortunately, with the heart, we haven't really been able to characterize what these stem cells are or really how to harness their power. Which raises a second possibility, and that's using the pluripotent stem cells, as has been mentioned, transforming them in the laboratory into a cell type that we might want to use, and then delivering them into the heart. And that's where the video on the right, that's heart muscle cells, cardiomyocytes, that have been created in my laboratory. And what you're seeing there is what I would see if I look down the microscope. That's a dish maybe about so big, and that contains maybe 10 million heart muscle cells that have been generated from stem cells, pluripotent stem cells. And you can see that they're robustly contracting their heart muscle, and we can now deliver that potentially to that failing heart that I was talking about. So in my laboratory, we're able to do that, make heart muscle cells by the billions now, and we're looking in animal models to find out ways to deliver them to our patients. So here's an animation, a nifty way of what we see might be the future. Heart attacks are caused by lack of blood flow to the heart muscle, and then the lack of blood flow causes death of that heart muscle. That's what you're seeing in the black. And you can see the heart's not beating so well anymore in the black areas, but look, we can come in with stem cells and we can inject the stem cells potentially and make everything better, right? Easy? Probably not so much. And that's why I'm introducing this concept of clinical trials, which we've already heard a little bit about. So this is a slide taken from the New South Wales government talking about cancer translation, but we can adapt it for our purposes here. So on the left of screen, we're talking about preclinical studies, and that's what we've heard so far. And in my research, it's firmly implanted at the moment in the preclinical area. And that does involve animal models, and in Australia, and in most places around the world, we very carefully choose about and respect the use of animals, rightly so. But I think animal models do have a place in improving that a novel therapy is safe before we go into delivering such therapies into my mother or my father or your grandparents. And then that's the stage of the phase 1, phase 2 clinical trials, where primarily we're focused on safety. Then once it's an accepted therapy, we treat patients, and then we can go into phase 3, phase 4 clinical trials where we're still refining the treatment. And that's at the stage where blood cancer treatments are at. We've heard that it's already started 50-odd years ago, but we're still doing clinical trials to refine and improve that therapy. So with that, I'll pass the clicker on, and thank you for your attention.
Just warning me not to trip. Okay, so I'm going to move to another organ and talk to you about the work that we do in the kidney. So your kidneys are really important organs. What your kidneys do is actually regulate how much fluid there is in your body, and they also filter your blood and remove any of the waste products that you actually don't want to do there, and of course they remove that from your body as urine. And it's essential that you have that renal function. You can't live without it. Unfortunately, in Australia, 1 in 10 people have some evidence that their kidneys are starting to fail, and so chronic kidney disease is actually increasing at about 7% per annum. Today, there's 13,000 Australians that are living only because they're on long-term dialysis, and there's more than 10,000 patients that are post-transplant having received an organ donor. Unfortunately, only 1 in 4 kidney disease patients will actually receive a transplant because it's very difficult to get one. So we've got a major problem here. We need to find some alternatives for treating kidney disease. So I've actually worked on kidney development for more than 25 years, and we use animal models to actually try and understand how you got your kidneys in the first place. How did this beautiful organ form? It's very complex, one of the most complex organs other than your brain in the body. It's many different cell types, and it has to make in a human kidney about 2 million little tubules called nephrons that are the points at which the blood is filtered and then collect the urine. What we know is, and you probably can't see this, the red tissue in the middle here, is the population of cells in the kidney that form the nephrons. These are the stem cells that actually have to grow, increase in number, and then turn into all the nephrons. And that's a really remarkable process. We know now the types of genes and signals that have to happen between cells for that to occur and for you to form a kidney. We know actually how to find them and how to characterize them. We know a lot about the different cell types and how they interact with each other. However, those stem cells are gone by birth. So the entire nephrons that you have in your kidney are formed before birth, and there isn't a stem cell able to make a new nephron after birth. So if you damage your kidney after birth, you haven't got any way of making new ones. What you're looking at here, however, is a kidney organoid. This is a model of a human kidney that we have built from pluripotent stem cells, which you've heard about, by essentially training those pluripotent stem cells to go through all of the steps that we know now occur during normal kidney development. So we've convinced them that it's kidney that they need to make, and it's actually formed a very complex organ with probably 15 different cell types that are making these tubular nephrons that would normally be present in a developing kidney. This was about 7 years of work in the laboratory, and it's quite, if you watch this movie up here, this is scanning from the bottom to the top. What you've got is a whole lot of little tubules connected to each other, and at the top of each there's these green balls of cells we call the glomerulus, and that's what we would normally see during development. Over in the bottom panel, the glomerulus is in red, and in the organoid they're in white. These are the cells that draw in the blood vessels that start to cause filtration to occur. Amazingly, not only do you have nephrons in this model, you have the cells around the nephrons, including these little spidery traces here in our grid of 4 to the top left, and this is the blood vessels forming. So you're getting blood vessels forming within the organ, and in fact, if we take one of these and transplant them into a mouse, the blood vessels from the mouse will grow into the organoid, and what you're looking at in the bottom here is green, a dye in the blood of the mouse, actually going in and going through the glomerulus of a blue glomerulus that we know is human because it's blue. We actually put a tag in there using gene editing. So we have a really beautiful model of a human kidney in a dish now. Of course, this model is only about 2 millimeters in diameter and has about 100 nephrons, whereas your kidney has 2 million. So what are we going to do with kidney organoids? How do we actually get something medically relevant out of this? Well, as I've explained, we're making these human models of the kidney out of induced pluripotent stem cells, and they can be made from any cell in your body, which means we can make stem cells out of a patient with kidney disease. And we're doing that. We're actually making iPS cell lines from patients with inherited forms of kidney disease. And this is actually an organoid, or kidney organoid, made from a patient line. When you zoom in, it's got nephrons in it, as you would expect. And this is a case in which, in this example, the patient had a disease gene. We'd sequenced that, and we felt that this was the gene that was causing the disease. And so we used CRISPR gene editing to fix the disease. And so we had a cell line from the patient and a cell line for the patient where we gene edited a correction. And when we zoom in on the cells, the little red dot there in the patient is short, and where it's longer when we've corrected. So we can show that that is causing the disease. But we can't always know exactly what's caused the disease. However, we can grow a model and we can screen drugs and start to ask, are there compounds that we can put onto these models of patient diseases that might be useful? To do that, you need large numbers of model kidneys, and so we're actually using this 3D printer to actually bioprint out large numbers of kidney organoids that we can then use in screening platforms, either to see if a drug works on that disease or to see if it's toxic to the kidney in some way. And we're hoping that we'll be able to look at both nephrotoxicity of new drugs, just to check in human tissue that they're not damaging, but also find new treatments for kidney disease, even if we don't know what the gene is that's causing that disease. Of course, in the long term, we actually hope that, given that we can make human kidney cells in a dish, that we'll be able to use those human kidney cells to do something therapeutic, either use those cells for cellular therapy in a similar way that James projected in the heart, or actually bioengineer a complex tissue. And as I've shown you, this is actually a little mouse kidney with a transplanted organoid on it. We have already started testing what happens when you move an organoid into an animal and shown that the blood will grow into this organoid. That's, of course, as I said, a much smaller tissue than what you would need. So again, this organoid is a couple of millimetres across. It's got 100 nephrons. You need 2 million nephrons to make a human kidney. So we've got challenges of scale, we've got challenges of structure, and we've got challenges of function, because what this is a model of a developing human tissue. We've got to actually get it to the point where it's functionally mature. And so that's a very long way away. Science is a slow process, unfortunately, and so we're a long way away from clinical trial, but hopefully moving towards it. And so I think I'll stop there. Thanks.
So you've just heard a fantastic series of presentations about how researchers are using stem cells in the lab now and how they hope to drive discoveries to make a difference to people. So I'm going to turn the microphone over to you now. Yes.
Hello everyone. So as part of my Year 12 course, I'm writing an extended essay on the use of stem cell treatments. And the thing that I'm focusing on is the comparison between the induced pluripotent stem cells and the embryonic stem cells and how reliable the methods are. And so I was wondering if you guys could explain a little bit more about the differences between them and why there might be different results for different types of stem cells.
Great question. Melissa?
I'm happy to have a go. I'm not sure if I'm on still, yeah. Both of those types of cells are what we call pluripotent. That just means we think they're able to turn into any other cell type. Embryonic stem cells were isolated 20 years ago. Making a pluripotent stem cell via reprogramming was only 10 years ago. And there was a lot of question when that happened about whether they were really equivalent and whether there were some subtle differences. And while there are some subtle differences, most methods where we try and turn a pluripotent stem cell into a tissue work with either. We routinely use embryonic stem cells or iPS cells, and we can make kidney out of it. And that's really the case for most of the protocols, whether you're making a heart muscle or blood or anything else. So they're really quite equivalent.
Is there a preference? So is one more effective than the other, or do you find that they're almost identical in how effective they are?
I find them almost identical, and we use almost exclusively induced pluripotent stem cells, because if I want to model a patient, I don't want an embryonic stem cell line, I want a cell line that's made from that patient. Now, you could take an embryonic stem cell line and try and put the mutation from your patient into that line, but most disease is not one gene, and so you really want the patient's entire genome in your line. And it is so straightforward to make an induced pluripotent stem cell line. We really can make it from pretty much any person.
Thank you very much.
Did anybody else on the panel want to add anything to that? Michael?
Yeah, I'll add one little thing. And Melissa's completely right. iPS cell technology is reproducible. Hundreds of labs around the world have used it and are able to use it all the time. But the little bit that I would add, though, is that there are couples who undergo IVF treatment because there's a family history of disease. And the embryos are screened to make sure that the embryo that's implanted doesn't have the disease that the family has a history of. And then the couples are left with the decision, what do they do with the embryos? Do they have them destroyed, or do they, if they wish, donate them for research to have that particular type of disease studied? And many couples choose to do that. So there is a source of disease-specific cells that you can get that people choose to donate to research because they want to make a difference in how their family is and the disease that affects them. So whilst iPS cells are certainly a new technology that's very reproducible and used by many, many groups and is a great way of getting disease-specific cells, there still is this place for embryonic stem cells.
Megan, I might just make a point about what I felt clinical translation would use first, and I was thinking that embryonic stem cells would definitely be where we get into the patients first. But we've already seen clinical trials started in the eye and in the heart using induced pluripotent stem cells. That's the reprogramming technique,
It's
which was
starting to
just
happen in the brain as well.
right,
Yeah.
which is just fascinating. I would never have thought that things would evolve so quickly into the clinical space. So I think iPSC will probably be used over embryonic stem cells clinically.
And again, I think just in terms of time frame, I mean, it's pretty remarkable that we're seeing those IPS cells, since the discovery was only 11 years ago, shifting into clinic. So that's, in our world, that's a really tight, really quick turnaround. Perhaps not, perhaps for some patients, but from our perspective, that's a remarkable series of events. Good luck with your assignment. I hope we helped. So there's a question over here and then one here.
I was wondering what advancements in technology you'd like to see in the near future that might help speed up or assist stem cell research.
Great question.
Because you said that 3D printing was available, and I assume you're 3D printing stem cells into a culture to allow them to grow. So is there anything more that can aid you in that process?
Pretender, do you want to?
Yeah, I was going to have a go at that. I think one of the limiting things for many applications is actually to have a vascularization, that is a blood supply, added to the tissue, because people are showing actually that if you graft a piece of skin or a bit of lung or whatever, which has already got vascularization made into it in the lab, that when you then transplant that, it anastomoses really quickly with the host supply, and the graft is going to take much better. And I think this is going to be complex for the different tissues we're all working with, but I think we can get that one down quickly, then I think that it'll accelerate our applications a lot.
Susie?
I'd like to add something to that. One of the huge advances that would be very helpful for us would be in visualization. So being able to actually see what's happening in the body in a bone setting would be fantastic. We've now got cameras you can swallow, they can see what happens in your digestive system. We've got all kinds of applications where you can visualise, but unfortunately, because bone is so dense, you can't see through it. You can in certain bones, such as the skull, but not in your bones where these stem cells are residing. So technology that would allow us to see through dense bone and visualise the stem cells in the body, in their environment, would be very advancing for us.
Actually, that's been a real game changer at the moment. So there are techniques like what Melissa was showing you, and I didn't bring any cool video of actually imaging whole human skin, where you have protocols which allow you to clear the tissue, and then you let antibodies bind to, molecules of interest on particular cells. And I was showing you actually blood vessel staining, which was actually pericytes, and then the epithelial cells, right? So I can look at the interrelationship, the placement of this cell versus this one, and I can say, is a stem cell more likely to be near a pericyte, for example? So I know that kind of what we've used for a long time, 2D imaging, cutting a section is a bit like trying to understand all of you by looking at the second row. But if we can look at all of you, or a good section of you, then we have a much better understanding of interrelationships within the tissue.
I think there's a question up the back.
Hi, everyone. My name is Ron, and this is Christina, and we are both medical students from the University of Melbourne. I just have a question in regards to the cardiomyocyte stem cell research. So I was wondering if there have been any research done into pacemaker cells and some of the other electrical cells within the heart, and kind of integrating that into the wider network of cardiomyocytes, having those pacemaker cells and having a more kind of broad understanding of the heart in terms of stem cell research? Thanks.
Great question. So yes, there has been, and pacemaker cells is what makes that heart muscle beat. But clinically, that's been one of the engineering feats of cardiovascular research, that we can use engineering devices to make the pacemakers block and heart failure. So stem cells have been looked at for pacemaker activity. There's been overexpression of a particular gene, I think it's TBX18, that can reprogram other cells into pacemaker-type cells. And as you've seen in that video that I showed, we already have within this pluripotent stem cell mix cells that have this pacemaker ability. So there are cells that we could use even as biological pacemakers within that mishmash that we've already programmed. Some of my research, an animal model, a large animal model, found cardiac rhythm abnormalities after we transplanted the heart muscle cells. And personally, although we remain to really understand it completely, I think it's those heart muscle cells acting as a biological pacemaker.
Can I just add to that, even though heart is not my primary field? I mean, as you've heard, this is a week where the international stem cell community is in Melbourne, and the meetings are already happening around that major meeting, which starts in a couple of days. And there's really a lot of new data coming out of being able to tell a pluripotent stem cell not just make a cardiomyocyte, but make one that's patterned to ventricle or patterned to atrium or patterned to the outer surface of the epicardium or patterned to the valve or patterned to the pacemaker. So we're getting better and better at actually more finely instructing the cells and then making models that are complicated, like the kidney one that I showed you, where you've got mixtures of the cardiomyocytes, the epicardium, the fibroblasts all together, which is essentially what a tissue needs. There's very few tissues that are made up of one cell type.
Hi, I'm Bonnie and I'm in year 11, and I just wanted to know, with so many options for what you can specialise in, why did you choose to specialise in stem cell research?
Who wants to go first?
Yeah, I'm happy to start. I won't say that I'm closest to high school age of anyone here, because actually I might be wrong, but for me it was experiments that were done in the 80s where they grew an ear on the back of a rat. And used that to transplant to a patient. And I was stunned by that. Just the idea, it was science fiction at the time. And so there's been this gradual progression from that captivating event through enjoyment. What have I liked? That's helped me specialise along the way. And then once you get into it and you realise what the potential is and you want to make a difference, then you bring all those things together, and it can be a very rewarding career. So something that captured my passion and then followed it through, kept it through. Yeah.
I was going to say that some of it is serendipity, actually. So I think when I was an undergraduate at university, I was going to do cancer research, right? And then as I searched for a PhD project, I ended up with this English gentleman who really was like the father of epithelial stem cell biology. And they were doing all these things in, animals, and having to think quite hard to draw conclusions from there about how this tissue replaced itself and that stem cells existed. And then I went off and did cancer research for a while, but I recognized that there was actually not a lot going on in this field as the whole stem cell area developed. And I'm happy to say I jumped on the bandwagon before stem cells became a big thing. So I think it was being exposed to cutting-edge research in that arena, and then seeing an opportunity that, yes, actually, there's no experts in this area, and I'm maybe just as qualified as anyone else, so let's give it a good go and make a contribution.
What about you, James? How come you went into stem cell research when you did a medical degree? You're a cardiologist. Why stem cells as well?
Yeah, I alluded to it in my talk that the heart being one of the least regenerative organs of the body. Although listening to Pratinder's talk, there's a lot of similarities. I thought the skin was very regenerative, but the heart can repair. It scars. But that scar still does have some contractile ability and some... Anyway, I digress. But the stem cells can be important for disease modelling, etc. But for me, it was the regenerative medicine aspects of stem cells. In medicine, we do a lot to patch up a little bit, to give small molecules drugs to fine-tune around the edges, but to really create new organs or to replace organs that were damaged, that's captivating, and that's why I'm in it.
So I've worked on kidney for 30 years, and I think that was an accident. Was just what I did my honours year on. But in 1998, two things happened in science. The first human embryonic stem cell was cloned, and Perry Bartlett published that your brain actually has stem cells, which I was taught when I was going through uni was not true, that you had neurons and then they died. And so that was really quite a revelation. And 2 years later, my very close colleague, who was also a developmental biologist, died tragically at 40. And I think it made me stop and wonder why I was doing fabulous and beautiful science, but not necessarily going to make a difference. And I really completely shifted what I was doing to work on whether what I was doing could be useful from a clinical point of view. And maybe one day I will make a difference. We hope so anyway.
And how about you, Susie? I mean, you're very close to the clinic. I mean, your area is already making a difference, but you're looking to take that a step further.
I guess similar to Melissa, when I started working this area many years ago, it was seeing that while medicine was great, there were so many gaps. There was so much that they didn't understand, and whilst they could do lots of things, there were so many things they couldn't do. And I really wanted to do something that could make a difference, that something I'd worked on would eventually get into the clinic and make a difference in people's lives.
Great. Well, I think that's a perfect segue, because I want to now invite Daniel Feller, who's in the audience. And we've heard a lot about the International Society for Stem Cell Research meeting this week, and it's in Melbourne, and we're really thrilled to have it. And we're going to be celebrating science. But what drives a lot of us is the difference that it will make to people's lives. And we're very lucky to have Daniel with us tonight. Daniel's actually part of the program. He'll be speaking on Friday afternoon to tell people a little bit about what stem cell research means to him and his family. So, Daniel, I was wondering if you do a little preview now and tell people why you and Holly are such strong supporters of stem cell research.
Thank you, Megan.
If you want to stand to address people, that would be fine.
Hello, everyone. I'm Daniel. This is Holly here. We're parents to young Harry, who's 7. And Harry was born with an ultra-rare syndrome called Usher syndrome that affects the ears, the hearing, and the eyes, and sight, and balance also. Historically, Usher syndrome was termed deaf blindness, which is totally devastating. However, with the onset of the cochlear implant, my son, who was born completely deaf, at 9 months old, was implanted with the cochlear implant created here in Melbourne. And now, at 7 years old, and actually a lot earlier than 7 years old, he can hear, speak, communicate perfectly well. So already the deaf side of deaf blindness is just not relevant. We found out 4 years ago that he had this syndrome, a genetic syndrome, that affects 4,000 people on the planet. Usher syndrome itself, all the forms of deaf blindness, affect 40,000, but his form of it, which is termed Usher syndrome type 1F, affects 4,000, so no one. And what it meant was that within, he would have been about 3-4 years old when we found out that he had Usher syndrome. Within about 10 years, he would start to lose his sight. And we very quickly became involved with the Centre for Eye Research here in Melbourne with Alex Hewitt and Alice Peebo, who were mentioned in Michael's speech earlier. And we now know that on account of stem cell research, the future has much, much more than hope. It's a reality that there will be treatments available within 3 to 4 years that, all being well, will arrest the sight loss that my son would suffer from. And how has this manifested itself? It's within stem cell research, when he was 4 years old, he had skin cells taken to do during a routine operation for grommets. Alex Hewitt came into the operation and took some skin cells from him, which have been regrown back from the pluripotent form into eye cells, into retina cells, I beg your pardon, sorry, into retina cells that are not only retina cells, they are cells with Usher syndrome 1F. This disease that my son has, this syndrome my son has, that affects 4,000 people in the world. So now the point is that thanks to stem cell research, experiments can be done in the laboratory on these cells that have been grown here in Melbourne and also in Hobart, I understand, that will make the treatment so much more efficacious and safe. And that is what it all comes down to, that there's hardly anyone in the world who would want to be the first in this, but because of stem cell research, we are able to have experiments done on retina cells that have been grown. And Michael made allusion to the ear that was grown on the back of the mouse, the world of science fiction you referred to it as. And that's exactly how we feel about this, that your world is coming to our world now. The future is here, thanks to stem cell research. At the very beginning, Professor MacDonald referred to this whole thing as an achievement of high science. And that is the other aspect of this, that it's not only stem cell research, it's the study of virus vectors that would deliver the CRISPR-Cas9 treatment. It's the bionics that have created the cochlear implants and dozens of other aspects and the work from laboratories and clinicians all over the world that have created this community that we're hugely involved with, of which stem cell research is the most evident in treatments that are going to happen for my son. So it's a small thank you to the people on the stage. Thank you.
Thank you, Daniel. So I fear our time is drawing near. I'm just going to make one comment and say I'm shocked to think that that ear stimulated you, because it slightly horrifies me. And one of the reasons it horrifies me is the sci-fi element and perhaps an expectation. I'm very concerned sometimes about how our science and perhaps other aspects of medical research presented to the public. And whilst that was a great example of engineering, it wasn't really growing a mouse. It wasn't regeneration like James has been talking about with the axolotl. It was actually shoving a scaffold under the skin and seeing whether it could sort of mould itself around. So I think it just is, I'm just going to make a comment that I think, again, you've got to be careful to interpret what sometimes is how the science is presented. And what some of the little mini organs that we're growing are not quite able to do that yet, even though the cartoonists do like to portray stem cell research as being able to open up an incubator and pull out a dish of little kidneys or left tibias or something. So unfortunately, we're not quite there yet for those things. We do have time for one more question, if anyone... Yes, sir. Helen?
I apologise if it's not the smartest question. I don't have a biological background, but I was just wondering, with stem cells, would it be possible one day to grow an organ which is even more superior to the one that we were born with? So, would a heart that's more resistant to various diseases and that sort of thing? So, a super being.
Fantastic question around enhancement, Melissa.
To make sure I'm on. That's a fantastic question.
Thank you.
Having just heard that we need to be careful about sci-fi, I think that the more sophisticated we get in terms of understanding what genes are doing in a cell to make a cell a heart cell versus a kidney cell versus a blood cell, etc., and the more able we are to actually engineer those genes to slightly alter them, we have the capacity now to start to make totally new cell types. Now, of course, that raises ethical challenges. But it is possible now to add genes, to take away genes, to change whole pathways in a cell. And there are some really amazing possibilities there. We heard about how it would be great if we could visualize. We will move very rapidly into a time where we can make a cell that's got a tracker in it that you can use your mobile phone to work out where it's gone. You know, to get a cell to take a payload to a destination, a protein that it may not normally have made, but you want to have taken to a particular location. These modified immune therapies where we're making new cell types to actually go and attack a particular target, these are now going to be the next generation. And so where we had gene therapy and there was a very slow start there and a lot of hiccups around delivery, and we've had stem cells, and that's been a bit of a low take-up too. They're coming together, and we will have gene-modified cells that do new things. That's where the future's going to change again. And so all of these are really amazing and challenging disruptive technologies, and we've all got to just be careful how we move forward in this space.
So the future of stem cells, synthetic biology is coming to you. Come to our next forum. We'll have a forum on that perhaps later in the year. Britina, do you want to make a comment?
Yeah, I was going to say not so much, a superhuman being, but certainly improving conditions, right, that you're born with and you don't have to live with. And the other thing I want to add is that we are getting a grip on what is changing as you age. So to age better, is definitely within our reach, I think, to try and understand what goes on in the cell interactions. We already have a handle on some of those things, at least in skin, in my lab, for example. So I think, cautiously, we're not going to make the bionic man, but I think we are going to have cure some diseases and age better, hopefully.
We got one more question.
One more question over. I'm just getting a microphone for you.
Thank you. Just on behalf of my wife, who's trying to overcome MS, just wanted to know if anyone can share about MS and stem cell treatment in Australia. We hear of a lot of people going abroad to Russia, to Germany, to Mexico, China, etc. And we've heard stories of some of our customers, some of our customers know people that have come back and it's been like 80% or 70% improvement, and there's some come back and there's been no improvement at all. They've actually been bedridden. We need to know, we'd like to know, what is the progress in Australia with stem cell treatment and MS in Australia? Can anyone, share with us?
So thank you so much for your question, and I think you're highlighting, the great challenge. So James showed that figure where he was talking about clinical trial, and I think that is my simple answer. Your question requires a much more complex response, but my simple first response is to say we've got to be careful. This is all experimentation. It's experimenting on you, and the safe way to do that, and I think the only way to do that, as part of a clinical trial. There has been a clinical trial undertaken for MS using hematopoietic stem cells, and that's being done in Sydney, and I'm going to get you some information about that trial. When things happen outside a clinical trial, and what we've seen a lot in the stem cell space, is clinics that perhaps don't have expertise in MS, who are trying to who will treat anybody, and quite cynically, I think as long as you have a checkbook, they will treat you. They may well believe that they have a solution. They may well firmly believe they're going to help you. But I think we've got to be careful of that kind of entrepreneurial, cavalier approach, and we must make sure that what's going to be put back in you has a solid scientific justification that it may work, and that it is as safe as we can make it. These interventions are highly invasive, and they do carry risk. I think we're getting the wind up on that. But thank you so much for raising the question, James. Would you like to make any other comment?
Yeah, firstly, thank you for sharing that story. And we've heard 2 examples of how stem cell technology and what we work towards can really make a difference. So, I think with MS, it's a relapsing and remitting disease often as well, and you have to take that into context. I'm not a neurologist, full disclosure there. And the cavalier mentality, I think, can be a little bit wrong, but that's also how we progress. The importance is that we do it safely and that we learn from them. So there's regulatory change in Japan and recently in the United States as well to maybe say how we develop new treatments might be a little bit wrong and maybe it needs expediting. Now, we're not sure how those changes are going to pan out, and the world is watching. But primarily safety and learning from the treatments and not being motivated by money, I think that they're important principles.
So thank you again for the question. I'm going to now do some more thank yous. And I first want to start with Graham. Professor MacDonald, thank you so much for your wonderful introduction and also for the support that the National Stem Cell Foundation has given this function, but also the stem cell community over now many years. We greatly appreciate it. Please join me in thanking Graham. To the fabulous panel, Susie, Michael, Pratinder, James and Melissa. What great leadership. Let's wish them all the very best in their research as they progress their discoveries. I also want to thank all of you for coming along, and particularly thank our young ambassadors who have donned those beautiful red lab coats, courtesy of MS Australia, in case you're wondering. And I think that they've been wonderful to give up their time and share their passion. These are the leaders, the future leaders of stem cell research and regenerative medicine in Australia, and I think we're in safe hands. We've got such a great group. I particularly want to thank Freya, Genevieve, Jenny and Rita, who were instrumental in bringing the group together. And again, thank you so much. Thank you to the fabulous high school students and their wonderful teachers and the Genome Technology Access Centre who helped them come along today. We've had some wonderful colleagues from Monash and Melbourne University, from the academic staff, who have been here to guide these students to talk about where, if they want to study this field, where they can do so at Monash and Melbourne. And I hope they've found it informative, and again, some great questions from these students. So again, we want to see you at university and look forward to it in the near future. So again, thank them. And I have one more. I want to finally thank my colleagues from the Australasian Society for Stem Cell Research and the International Society for Stem Cell Research for supporting this meeting, but also for coming to Melbourne to celebrate stem cells. Finally, Julia Mason from National Stem Cell Foundation and Helen Braybrook, my colleague from Stem Cells Australia. They have been a tower of strength in pulling tonight off, and I want to thank them so much for their support. If you are interested in stem cell research, which you obviously are, and probably even more so after such an extraordinary discussion and panel tonight, we want you to sign up for our newsletters. You can find out more information on the National Stem Cell Foundation of Australia's website. You can look up Australasian Society for Stem Cell Research or Stem Cells Australia. But you could also visit A Closer Look at Stem Cells, which is the International Society's website that has been tailored for the community. So please hop online, stay in touch, stay informed, and come along to our next forum. Thank you so much.